as you've already heard, and probably all of you are aware, there's overwhelming evidence now that there are all sorts of non-genetic processes that contribute to heritable variation. But we still have a really poor rudimentary understanding of the role of these kinds of processes in evolution. And that's what I'll talk about here. I'll talk about a few studies that I think bear on this question and suggest in particular a way that these kinds of processes can play a role in sexual coevolution. I'll use that as a kind of model. So, for me, the most interesting aspect of non-genetic inheritance, including epigenetic inheritance, is the potential for it to transmit environmental variation across generations. For me, this is the way that it differs most from the classic kind of genetic, Mendelian genetic model. And if we think about this potential to transfer environmental variation, I think we need to go back a couple of centuries to the writings of Lamarck. And he suggested, he wrote a book suggesting that environmental variation can be transferred across generations. He based a theory of evolution on this idea. And he also believed that this variation was adaptive. And the big hole in his writing is that he never provided a mechanism to explain how this variation might spontaneously be adaptive. A few decades later, Darwin built on Lamarck's ideas. Darwin also believed that the environment generates heritable variation. But Darwin suggested that the environment also imposes natural selection in this variation, and that's how adaptation comes about. And by the middle of the 20th century, Darwin's theory of natural selection was reconciled with the new field of genetics to produce the evolutionary modern synthesis, which is based around the idea that the environment generates natural selection on genes, genetic variation. And the modern synthesis very, very clearly, explicitly repudiates or rejects the idea that the environment generates heritable variation. So where are we now, several decades later? Well, if we're interested in the role of environment in generating heritable variation and the potential for this to influence evolution, I think a nice place to look is sexual coevolution, in particular condition-dependent secondary sexual traits, because these traits are famously sensitive to the environment. And there's also quite a lot of theory built up over the past several decades on how these kinds of traits may influence sexual coevolution. In particular, the idea that these kind of traits will tend to signal genetic quality, and that females will evolve to prefer males that express really nice secondary sexual traits. And the most explicit formulation of this idea is Rowan Poole's genic capture model. So according to this idea, the environment contains resources, and organisms attempt to extract resources from the environment to build up a kind of pool of metabolic resources that they can use to invest in all of their various fitness-enhancing functions. And one of the key functions that they invest in is the expression of secondary sexual traits, like signals. And females are then able to choose males based on these signals, because these signals then reflect the quality of genes throughout the genome that determine performance at extracting resources from the environment. This model is almost 2 decades old, and it's been incredibly influential. So from classic theory, the modern synthesis and the Roland Poole model, we have a couple of premises that are interesting to kind of re-examine in light of what we know of more recent evidence. Firstly, that condition-dependent secondary sexual traits actually serve as really good signals of genetic quality. And secondly, that environmental variation in condition is non-heritable. It's noise, as it would be formulated in a classic quantitative genetics model. So I'll tell you about some studies recently that I've been involved in that bear on these premises or assumptions. So firstly, in collaboration with Howard Rundle and his group and some statisticians in Spain, we wanted to directly compare compare the role of genetic and environmental quality in influencing trait expression. And we chose this wonderful organism, Drosophila melanogaster, and to do this, we used a set of 19 hemiclones, which are haploid genomes, and each of these hemiclones had been propagated for 50 generations, either under a mutation accumulation protocol, where the hemiclone was propagated for a single copy, presumably allowing deleterious mutations to accumulate, or through controlled conditions where individuals could compete, and presumably selection would filter out lots of deleterious mutations. So this was a genetic quality manipulation. And then each of these hemiclones is reared on either a high-quality larval diet or a low-quality larval diet, which is an environmental quality manipulation. And so this is a fairly severe genetic quality manipulation. This was a fairly mild environmental quality manipulation. I just want to show you one set of results from this. So this is from male morphological traits. There's a bunch of different traits here. This is a plot of the effect of environmental quality on trait expression versus the effect of genetic quality on trait expression. And this is the line of equality here. And as you can see, the point estimates for all of these traits are greater for environmental quality, which is with the exception of this one. But I want to draw your attention to just one trait, which is the male sex tones. And this is considered a classic secondary sexual signaling trait in Drosophila melanogaster. It's a set of highly modified bristles on the foreleg, which are used by males to stimulate females, and there are studies suggesting that the sex tones are under sexual selection, we'll say. What we found was that there was a strong signal of environmental quality on the expression of this trait, but there was absolutely no signal of genetic quality, no consistent signal whatsoever. The estimate was pretty much zero. So here's a highly condition-dependent secondary sexual trait that is absolutely useless as a signal of genetic quality, which I thought was kind of interesting. So, okay, so it signals environmental quality. Many of these traits seem to signal environmental quality. Can that have any implications in sexual coevolution? Well, in my group, we've been working for a number of years on these lovely Australian Reapplies, Telestelinas angusticolis, which are interesting in being extremely plastic, extremely sensitive to environment, in that if we rear them on a low-quality larval diet, these are two males, they're two brothers actually. Males come out very small and shaped very much like females, with short legs, short stubby heads, and antennae. If we rear them on a high-quality larval diet, males are much larger than females, and they express exaggerated secondary sexual traits like elongated heads and antennae. We also found a few years ago that if we manipulate larval diet in this way, and we take these different phenotypes, and we pair them with standard mates, we see a paternal effect on offspring body size. So males reared on a low-quality larval diet produce smaller offspring than males reared on a high-quality larval diet. So this is a way that males transfer their environmentally acquired condition to their offspring. More recently, we also found that males can transfer their environmentally acquired condition to offspring sired by other males that later mate with the same female. So in this experiment, I should go back and say that here we surmised that these effects may be mediated by substances in the seminal fluid. So we wanted to see whether we could actually decouple this non-genetic paternal effect, this non-genetic effect from fertilization. So females in this second experiment were paired in succession with two different males reared on either the same or different larval diets. And the first mating took place when females had immature eggs, which we thought would be more susceptible to the non-genetic influence. And 2 weeks later, females were paired with another male once their eggs had matured. And we predicted that that second male would actually fertilize the eggs. And what we found was that indeed most of the offspring were sired by the second male. So this is based on offspring sired by the second male. But it's the first male that affected the body size of the offspring. The first male's acquired condition determined the body size of the offspring. So this is kind of an interesting non-parental, non-genetic effect that shows that, that this kind of non-genetic transfer of condition is clearly decoupled from any sort of genetic variation. And this effect is decoupled from fertilization, so there's no potential for genetic and non-genetic variation to be kind of transmitted together. So what about sexual coevolution? Well, Troy Day and I thought about whether these kinds of effects can play a similar role to genetic variation in fitness in driving sexual coevolution. So we built a kind of simple population genetic model, except that we designed it so that male condition was determined essentially by luck, by the environment in which a male grew up, which was determined entirely stochastically. So some males would grow up in a high quality environment, and they would be in high condition. Other males would grow up in a low quality environment and be in low condition, and males can transfer this condition then to their offspring. Females have a locus which determines preference. They could either prefer males in high condition, or they can be indiscriminate. What we found was that this kind of a paternal effect readily drives the evolution of costly female preference, just like in a good genes model, except that this is very, very different from a good genes model. Here, fitness is determined by paternal environment. And of course, this kind of a system avoids a classic problem with good genes models, which is the Leck paradox. Heritable variation in fitness can't be depleted here because it's regenerated every generation by the environment. So, how much time do I have? 2 minutes. Okay, so what about secondary sexual traits themselves? Well, given what I've told you, it's interesting to ask whether actually secondary sexual traits might evolve to capture environmental variation in condition. So we might call this environment capture. Perhaps secondary sexual traits actually evolve to signal environmental variation in condition. Females can choose males based on these traits because they get beneficial paternal effects for their offspring. And this is, if this actually occurs in some traits at least, it's kind of interesting because, firstly, these kind of traits also work very nicely in intrasexual signaling. Males don't care about the genetic quality of their rivals, only their phenotype. This avoids the Leck paradox, and this also avoids potentially the problem of sexually antagonistic fitness variation. So perhaps, rather than looking for a signal of genetic quality, in our studies of sexual coevolution, maybe we should simply accept that environmentally generated variation in condition can play a similar role, a similarly interesting role in sexual coevolution. So to summarize, I think there's good reason to believe that environmentally generated variation in condition could influence sexual coevolution, both because it can be transferred across generations paternally, and this can influence the evolution of female preference, and potentially secondary sexual traits may actually evolve to signal environmental variation in condition. That's it. Thank you.
It's time for questions. Anyone? No? Okay, so you've shown that